Good afternoon. My name is Bill. On behalf of my collaborator, Xianming Li, I would like to introduce our drug carriers, which we believe have the potential to transform drug delivery in today's cancer therapy.
It is becoming increasingly clear that the enhanced permeability and retention effect, also known as the EPR effect, is no longer a reliable mechanism for effective drug delivery to tumors. Several landmark studies have demonstrated that the delivery efficiency of nanoparticles to tumors is only about 0.7%. Unfortunately, this has a significant negative impact on the therapeutic outcomes of many anti-cancer drugs. Often, drugs fail not because they are ineffective, but because they never reach the target sites, leading instead to toxicity in healthy tissues. As you can imagine, this issue not only results in millions of dollars in losses for pharmaceutical companies, but also has a profound impact on the survival and quality of life of cancer patients. One of the key pathological events in cancer is the recruitment of monocytes to tumor sites. These circulating cells have a strong natural homing ability towards tumors. Based on this, we have developed peptide liposomes like an anchor to the surfaces of circulating monocytes. We call these drug carriers as monocyte-mediated drug carriers, or MMDCs. Once introduced into the bloodstreams, MMDCs will hitchhike by anchoring on the circulating monocytes. These monocytes then transport the MMDCs directly to the tumors and not to the healthy tissues. Panel B shows that our MMDCs labeled in red can anchor on the surfaces of circulating monocytes, shown in green. In contrast, the control liposomes, which are the classical pegylated liposomes, fail to anchor onto the monocytes. Panel C shows the hitchhiking efficiency of MMDCs in a xenograft mouse model of breast cancer. Only about 2% of control liposomes remain anchored on the circulating monocytes, while MMDCs show the hitchhiking efficiency close to 50%. Panel D shows the MMDCs enhance tumor targeting by at least tenfold compared to the control liposome. Finally, Panel E shows that the mice treated with MMDC encapsulated doxorubicin had the smallest tumor sizes compared to all other treatment groups. With the funding support from this proposal, we plan to demonstrate the therapeutic efficacy of MMDCs in delivering gemcitabine in PDX models of pancreatic and colon cancers. It has been demonstrated that monocyte recruitment is closely associated with the progression of both types of cancer. Furthermore, the demand for new target therapies for both cancers is significant. The global market outlook for pancreatic cancer is projected to reach approximately $6 billion by 2030 and $25 billion for colon cancer.
Our ultimate goal is to demonstrate that MMDCs can address complications associated with drug interactions in patients with multimorbidity. In Taiwan, 70% of elderly individuals have multimorbidity. Among them, 85.6% are experienced with at least one complication related to drug interactions. We plan to develop specialized MMDCs, where one batch designed to recognize monocyte recruit to the infant heart tissue, for example, and the other batch designed to target another disease, for example, like tumors, where the drug carriers, MMDCs, will recognize that specific monocyte populations that are recruited to the tumors. This way, patients suffering from both conditions can be treated without worrying about drug interaction when receiving MMDC encapsulated therapies. Our first generation MMDCs have already been patented in many countries. We are currently collecting additional data to support the patent application for our second generation MMDCs. Additionally, we anticipate to submitting more patent applications after optimizing the formulation of MMDCs for different chronic diseases. While other cell-mediated drug delivery systems do exist, we are confident that our MMDCs are superior. As mentioned earlier, our MMDCs are designed to anchor on the surfaces of circulating monocytes under high shear stress environments. And these drug carriers are only internalized by the cells after the monocyte had differentiated into macrophages. In contrast, the design proposed by others are sometimes in fact too bulky, as shown in this diagram here, which can hinder the cells undergo transendothelial migration. Whereas some of the cell-mediated drug carriers in fact require to premix with the cell before introduced back into circulation. The downside of doing so is in fact the premixing can in fact lead to premature drug release, which can lead to undesirable side effects. Whereas some of the design can in fact lead to self-aggregation while in circulation, which can lead to undesirable blood clotting.
Our primary commercial strategy at the moment is to license out the MMDC formulation to pharmaceutical companies in Taiwan that are interested in repurposing their drug candidates. We are already working with some of the pharmaceutical companies right now in Taichung, where we're assisting with some of the drug repurposing experiments. At the same time, we hope to establish our own therapeutic products by collaborating with research groups that have promising drug candidates but face poor efficacy due to inefficient drug delivery. At the moment, we are working with a group in Germany where we are optimizing our MMDC formulations so that we can encapsulate their nucleic acid-based drugs for treating patients with myocardial infarction. Currently, in our team, Shen Ming and I are the key drivers of the MMDC technology. We are continuously developing and optimizing formulations for various chronic diseases. We also have Dr. Jingjong Zhang on our team, who has strong connections with pharmaceutical companies in Taiwan, China, and several Southeast Asian countries. Dr. Zhang is currently assisting us with commercial licensing efforts. Another key player of our team is Dr. Victor Guo. He's the head of the Oncology Division at Guangtian Hospital. Dr. Guo has been providing valuable clinical guidance, particularly regarding the experimental data needed to support future clinical trials. Our MMDC technology has received recognition from the committee of Moderna Taiwan mRNA Innovation Award, where we will select one of the finalists for both 2023 and 2024. We also won the Best Presentation Award at the 40th Annual Meeting of Japan Society of Drug Delivery Systems, as well as the Young Investigator competition at the annual meeting of the Biomaterials and Control Release Society in Taiwan. With that, this concludes my presentation. I'd be more than happy to take any questions. Thank you.